This is the teacher tip video for fermenting fuel, designing a procedure for fast fermentation. Ethanol is a renewable fuel source produced by the anaerobic fermentation of sugars by yeast. As you can see in the equation below, one glucose molecule is used to produce 2 ethanol and 2 carbon dioxide molecules. This is a 2:2 ratio, which simplifies to 1:1, meaning that for every molecule of ethanol produced, a molecule of carbon dioxide is produced. The yeast uses the energy stored in the 2 ATP molecules. We will collect and measure the carbon dioxide gases produced in order to compare rates of fermentation. Yeast is very good at metabolizing simple sugars. The sugar in the corn plant is in the form of starch, which needs to be broken down. Enzymes are used to break the starch molecule down into smaller pieces. Amylase just makes shorter chains. Glucoamylase releases free glucose molecules. This lab was designed to allow students to test many different variables in an effort to produce a faster, more efficient procedure for fermentation. Varieties and amounts of yeast can be tested, different combinations of enzymes, changes in pH, temperature, or even the time of heating and preparing the sample. Each lab group will need 5 samples, and each sample will need 1 test tube, 1 gram of ground corn, roughly 5 milliliters of distilled water, a stirring rod, and a test tube holder. First, add the 1 gram of ground corn to each test tube. Add approximately 5 milliliters of water, then use a stirring rod to mix it well. You also need to prepare a water bath. Boiling breaks down the cellulose in the cell walls and releases the starch into solution. Add some water to a 500 milliliter beaker and bring it to a boil on a hot plate. Using test tube holders, add your samples to the boiling water and let them boil for 10 minutes before removing them. It is important to make sure your samples have cooled before adding enzymes, as heat will denature them and prevent them from working. Samples can be stored for the next day if you put a stopper in them. This is an example of a student-developed procedure. You can see in the first row they will be adding amylase, and they have marked how much will go into each test tube. 1 milliliter in test tubes 2 and 3, and test tube 4 receiving 2 milliliters. Test tube 1 and 5 will not have any amylase added. You can see in the next row on the table they've decided to put 1 milliliter of glucoamylase in test tubes 2 and 3, 2 milliliters in test tube 5, test tubes 1 and 4 do not get any glucoamylase. The next row shows that all of the test tubes except test tube 3 get 1 milliliter of pH buffer, and all of the samples will get 1 gram of Red Star Distiller's Active Dry Yeast. They will ferment in a 30 degrees Celsius water bath. Distilled water can be added to equalize the volume of the samples. At this point, you will want to stir to thoroughly mix the samples. Test tubes will then be placed in a test tube rack, and 1-hole rubber stoppers with tubing will be inserted. The tubing will go into a manifold used to collect the carbon dioxide gas. To set the manifold, fill the tub with water, submerge the manifold and tip it over on its back. Tap several times to remove the air bubbles and tip it up. It is now ready to insert the tubes. As carbon dioxide bubbles are produced, they will be collected in the centrifuge tubes, and the graduated marks will allow you to collect data. This is a time-lapse video of a student's experiment. At the end, you'll notice the tubes start to slide around a little bit. To prevent this, you can put a piece of tape across the tubes to hold them down. If you're having trouble seeing the water, add some food coloring to make the water more visible. Collect and record data every 5 minutes and graph the data so you can compare your test tubes. In this case, test tube 3 produced the most carbon dioxide. Referring back to the student's procedure, we can see that test tube 3 was the test tube that received 1 milliliter of amylase, 1 of glucoamylase, and no pH buffer. You may want to allow students to run several different experiments so they can design their own procedure for a larger scale fermentation to see who can produce the most ethanol. I plan to give my students costs for enzymes, heating, different types of yeast, so they can plan a procedure thinking about how they can produce the most ethanol for the least procedural cost. The student-developed procedures can then be used in the corn mash and distillation lab, where students can compete to produce and distill the most ethanol. Now I'll go over a few tips for setting up your equipment that was provided by Seed to Stem. In your tote, you should find 4 3D printed manifolds. The PLA that these were produced from is manufactured using cornstarch. You should also find 25 50 milliliter centrifuge tubes. Remove the lid from the centrifuge tube and carefully thread it into the manifold. Repeat this 4 more times until all of your centrifuge tubes are in place. Try to make them as straight as possible. These have been numbered so students won't confuse their data points. Also in your totes, you should find 20 pieces of clear tubing. Determine the size of rubber stoppers you need to fit the test tubes you will be using, and then insert the tubing into the rubber stopper. If you have trouble fitting the tubing, use a pencil to widen the opening, and then try again. To speed things up on day 2, I have students pre-measure the amounts of yeast that they will be using in each sample, and store them in a 15 milliliter centrifuge tube. The next day they can just simply dump them in after they've used their serological pipettes to add their enzymes and buffer solution. At the workshop, we used water baths to control the temperature. I know water baths will be limited in your lab if you have one at all. In the picture, you can see a setup that we used to control the temperature without using a water bath. Students added warm water to the tub on the left. When their water cooled off, they simply scooped some out and added some more hot water. They kept their temperature range between 38 and 40 degrees. The 2 test tubes on the right were room temperature. When finished with the lab, I have students run water through the tubing and then store everything in their Ziploc tubs so the lab will be ready to set up the next time I want to do it.